Dear students, in this video we will talk about the dark reaction of photosynthesis. It is also called Calvin cycle, or it may be called C3 cycle. Okay, so in this process, what happened? In this process, CO2 reacts with ribulose 1,5-bisphosphate in the stroma of the chloroplast, and as a result of that, product synthesize. What is the product? The product is glucose, sucrose, etc. And it is a cyclic process by which ribulose 1,5-bisphosphate is again resynthesized or regenerate. And on the basis of the different types of reactions, the whole process is divided into 4 phases, and these are carboxylation, reduction, regeneration, and product synthesis. Okay, let us see. First, we will discuss about carboxylation, because carbon dioxide is fixed here. Okay, so let us start. Carbon dioxide reacts with 5-carbon ribulose 1,5-bisphosphate in the presence of the enzyme Rubisco, and as a result of that, one unstable 6-carbon compound is formed, and the compound is diphosphoorbital, or we may say DPOB. Soon the formation of this unstable product, it reacts with water. Okay, this unstable product reacts with water in presence of Rubisco to produce stable 3-phosphoglyceric acid. This one. Since 3-phosphoglyceric acid is 3-carbon and stable one, so this cycle is also called C3 cycle. Okay, that is 5-carbon ribulose 1,5-bisphosphate reacts with carbon dioxide to form 6-carbon unstable diphosphoorbital. This unstable product reacts with water to form 2 molecules of stable 3-phosphoglyceric acid, which are 3-carbon. Hence, it is called C3 cycle. Okay. In the next step, ATP is utilized, and here the process is governed by the enzyme phosphoglycerokinase. So ATP donates phosphate to 3-phosphoglyceric acid. As a result of, ATP is converted to ADP, and 3-phosphoglyceric acid is converted to 1,3-bisphosphoglyceric acid. That is, 2 molecules of 3-phosphoglyceric acids will be converted to 1,3-bisphosphoglyceric acid, again 2 molecules, in the presence of enzyme phosphoglycerokinase with the help of ATP, which were produced in the light phase. Clear? Okay. Then we will take a look on the reduction process. Okay, or we will see how reduction takes place. So our product is 1,3-bisphosphoglyceric acid, and this product reacts with NADPH of the product of light phase. Okay, and as a result of that, NADP positive ion, HOPO3 negative ion and glyceraldehyde 3-phosphates are formed, and the whole process is regulated by or catalyzed by the enzyme 3-PGA dehydrogenase. Okay, that is, we may say that 1,3-bisphosphoglyceric acid reduced to glyceraldehyde 3-phosphate with the help of NADPH, and the reaction is catalyzed by 3-PGA dehydrogenase enzyme. Okay, let us see what happened then. In the next phase, we will discuss about product synthesis. Okay, so take glyceraldehyde 3-phosphate, this one. It will be converted to dihydroxyacetone phosphate in the presence of the enzyme triose phosphate isomerase. That is, 3-phosphoglyceraldehyde is isomerized to dihydroxyacetone phosphate. Okay, this dihydroxyacetone phosphate then reacts with 3-PGALD. Okay, so 3 carbon plus 3 carbon, it is 6 carbon. So the result will be 6 carbon fructose 1,6-bisphosphate. 3 carbon from here, 3 carbon from here, 1 phosphate from here and another phosphate from there. That is why 6 carbon and 2 phosphate product fructose 1,6-bisphosphate, and here the enzyme is aldolase. Okay, next, fructose 1,6-bisphosphate is converted to fructose 6-phosphate by removing 1 phosphate which was attached to the first carbon, and here the enzyme is fructose 1,6-bisphosphatase and the product is fructose 6-phosphate. Clear? Okay, again I am telling, fructose 1,6-bisphosphate will lose the phosphate attached to the first carbon, and here water will be utilized, and as a result of that fructose 6-phosphate will be produced and the whole process will be catalyzed by the enzyme fructose 1,6-bisphosphatase. Then fructose 6-phosphate will be converted to glucose 6-phosphate, then to glucose 1-phosphate and ultimately glucose. Okay, this is the product synthesis process. Okay, now we have seen that food material, that is glucose C6H12O6, is produced in the process of Dulc reaction. by the utilization of ribulose 1,5-bisphosphate. Now the question is the regeneration of ribulose 1,5-biphosphate or bisphosphate. Okay, at first 3-PgALD will be converted to dihydroxyacetone phosphate, and dihydroxyacetone phosphate will react with 3-PgALD in the presence of the enzyme aldolase to produce 6-carbon fructose 1,6-biphosphate. Up to this we have seen in the previous product synthesis process also. Okay. After the formation of fructose 1,6-biphosphate, it will be converted to fructose 6-phosphate. Okay. So this fructose 6-phosphate will react with 3-PgALD in the presence of the enzyme transketolase. So 6 carbon and 3 carbon, total carbon is 9. So these 9 carbons and 2 phosphates, they will be divided into 2 products. One is 5 carbon xylulose 5-phosphate, another one is 4 carbon erythrose 4-phosphate. Okay, and the enzyme is transketolase. Let us see what happened to the 5 carbon xylulose 5-phosphate. This 5 carbon product is converted to ribulose 5-phosphate in the presence of the enzyme epimerase. Okay, then ribulose 5-phosphate will be converted to ribulose 1,5-bisphosphate by the utilization of phosphate from ATP in the presence of the enzyme phospho-ribulose kinase to produce ribulose 1,5-biphosphate again. That is ribulose 1,5-biphosphate or RUBP is regenerated from xylulose 5-phosphate through ribulose 5-phosphate. Clear? Okay. Now let us see what happened to the 4 carbon compound erythrose 4-phosphate. This 4 carbon compound, it reacts with DHAP, which was the product of PgALD, in the presence of the enzyme aldolase. Okay, as a result of that, 4 carbon and 3 carbon, they combine together to form 7 carbon compound with 2 phosphates, that is called shadow haptolose 1,7-bisphosphate. Okay, so erythrose 4-phosphate and dihydroxyacetone phosphate combine together to form shadow haptolose 1,7-bisphosphate in the presence of enzyme aldolase. And in the next step, this shadow haptolose 1,7-bisphosphate is converted to shadow haptolose 7-phosphate by removing 1 phosphate. Here the phosphate receiver is water, and as a result of that, shadow haptolose 7-phosphate is formed. That is 4 carbon erythrose 4-phosphate is first converted to shadow haptolose 1,7-bisphosphate by the utilization of DHAP. And in the next step, shadow haptolose 1,7-bisphosphate is converted to shadow haptolose 7-phosphate. Okay, so a 7-carbon product is formed having 1 phosphate. So our aim is to 5-carbon product, but here it is 7-carbon. Hence, again this 7-carbon product will react with 3-PGALD in the presence of the enzyme transketolase. As a result of that, 2 5-carbon products are formed. One is xylulose 5-phosphate, another one is ribose 5-phosphate. Eventually, xylulose 5-phosphate will be converted to ribulose 5-phosphate by the enzyme of epimerase. And again, ribulose 5-phosphate will be converted to RUBP by the utilization of ATP in the presence of enzyme phosphoryburose kinase. Okay. So, what is the remaining product? Or we may say that we have now ribose 5-phosphate. So, isomerization will take place. This ribose 5-phosphate will be converted to ribulose 5-phosphate in the presence of the enzyme isomerase, and ultimately again this one will be converted to RUBP by the utilization of ATP in the presence of the enzyme phosphoryburose kinase. Okay. So this is about the regeneration of RUBP. Clear? Again, I am telling in a nutshell. 3-PGALD first converted to DHAP, and DHAP is reacts with 3-PGALD to form fructose 1,6-bisphosphate. This fructose 1,6-bisphosphate will be converted to fructose 6-phosphate. Then fructose 6-phosphate will react with 3-PGALD, and in their combination 9 carbons are resulted. These 9 carbons are divided into 2 products. One is as xylulose 5-phosphate and another one is erythrose 4-phosphate. Xylulose 5-phosphate is converted to ribulose 5-phosphate by the enzyme epimerase, and ribulose 5-phosphate will be converted to RUBP with the help of ATP in the presence of enzyme phosphoryburose kinase. Okay. Then what happened to this 4-carbon compound? This 4-carbon compound again reacts with 3-carbon DHAP, okay, dihydroxyacetone phosphate, and as a result of 7-carbon product is formed, and it is none other than shadow haptolose 1,7-biphosphate. This shadow haptolose 1,7-biphosphate will be converted to shadow haptolose 7-phosphate by removing 1 phosphate, and this 7-carbon product will react with 3-carbon PGALD in the presence of the enzyme transketolase. As a result of that, those 10 carbons will be divided into 5-carbon xylulose 5-phosphate and ribose 5-phosphate. We have seen that xylulose 5-phosphate is converted to ribulose 5-phosphate by the enzyme of epimerase, and then again to the RUBP by the activity of the enzyme phosphoryburose kinase. So the remaining 5-carbon ribose 5-phosphate will be isomerized to this one ribulose 5-phosphate, and at the last this ribulose 5-phosphate will be converted into RUBP. Okay, so we have started from here, again we return back this one at this point, that is there is no displacement. Okay, in the term of physics, hence it is called a cyclic process, and by this cyclic process RUBP is regenerated, and during this process glucose was produced as a product synthesis, which was discussed at the very beginning. Okay, thank you.